Good morning. I am here to give you a quick lecture on lesson 2 for microbiology. So this is, well, it's almost midweek. By the end of the week, you should have completed most or all of your work, and everything is due on Sunday night. So it is now Thursday morning. So please make sure you have done your discussion post, which was due yesterday, and making sure that you do all your assignments through Sunday. So let's get started on the study of medical microbiology, lesson 2. This is the PowerPoint from the book. As you know, I kind of go off on tangents, so I'll try to keep it short and simple. I definitely recommend that you read the book. This is by no means complete. This is just the highlights. So make sure you read the book, do the PowerPoints, watch the learning videos, and basically take all the quizzes that you have with respect to your ebook. All right, so medical microbiology, what is it? It is the study and identification of organisms that can be potentially pathogenic, and how do we manage it medically. So it is not just an emerging field, it has been an established field that is very diverse, and it doesn't just include one or two specialties. It has numerous specialties within an umbrella called medical microbiology, which includes, of course, the study of bacteria. So there's people that simply just study bacteria and how to manage bacteria with antibiotic therapy. And then there's virologists, mycology, which is the study of fungus, and parasitology. So we're going to go over each one of these specialties within the umbrella of medical microbiology, with each chapter focusing on one of these branches. So this is only, the beginning, as I say, the foundational points that we are beginning to build upon. And so by week 5, you will start to learn more about all of these branches. All right, so with that said, we have a term to learn. So the prevention of these diseases and associated infections, the term is HAIs, right? Health-associated infections and healthcare-associated infections, I should say, is basically nosocomial infections, or infections that are unfortunately caught in the healthcare system when you are in the care of a multitude of practitioners in a hospital setting. That's called a nosocomial infection. And unfortunately, it is more common than you think, and it is associated with the lack of cleanliness in the hospital, not necessarily the hospital itself, but the employees. And so you being a future employee of a hospital, it is extremely important to understand not just sterilization, but also hand washing. Hand washing is separate from the sterilization process. Scrubbing the hands for 15 minutes prior to going into the OR, that's a whole separate thing. But frequent hand washing between seeing patients on the floor, in the clinic, these are habits that begin and are foundational, begin at this level when we are educating you prior to you entering any kind of clinical setting. Now, let's go over the history of microbiology. The history of microbiology begins with Anthony van, let me see if I can pronounce this correctly, Leeuwenhoek. Leeuwenhoek. All right, I'm not German, but I know that is German, Swedish. It is definitely, or maybe Scottish, because look at what it says, "we beasties." So he was able to view the organisms And so he named them wee beasties. So wee sounds a very Scottish term. But at any rate, he is the first one to have discovered organisms in the 1600 to 1670s. Present day, we have microscopes to view microbes, everything and anything from bacteria, viruses, prions, parasites, and fungi. All right, so there are numerous classifications of antibiotics for bacteria. I want to really delineate that because even in my previous classes, there were people that didn't know this. There are antivirals, that not a lot of antivirals out there, that inhibit the growth of viruses, but we really don't have much against viruses, okay? But we do have antibiotic therapy, thanks to the discovery of penicillin. And from there, the world opened up and we discovered, coming up with different types of antibiotics as the new strains, bacterial strains mutated, we came up with different antibiotics. Okay, so antibiotics are for bacteria. Vaccines is what we developed to fight the viral infections that are out there. Unfortunately, we don't have some pill per se. We do have some things that kill viruses, but not as extensive as antibiotic therapy against bacteria. All right, so the science of microbiology is basically developing a theory. The scientific method. We've probably gone over it in different classes that you have taken when it comes to the scientific method. Scientific inquiry is a very planned and deliberate investigation, developing a hypothesis, and then creating an experiment that will support your hypothesis. So scientific theory is based on information that is gathered from the natural and physical phenomena observed. And then the test must be replicated again and again with unaffiliated researchers. So just because you came up with the result, that result, for it to become a theory that is accepted, the results need to be replicated across other scientific labs. Okay, and not hearsay or saying it is said that if you do this, it results in that. Well, who said it? You need to prove it, and it needs to be a replicable experiment. Okay. Now, let's talk about bacteria and the study of bacteriology. The study of bacteriology is really involving not just the genetics, but how it reproduces and how we can stop the reproduction. So that's super important in terms of antibiotic therapy. So just so you know, bacteria, the way they reproduce is binary fusion. The process of bacterial reproduction is called binary fusion, where the cell copies its DNA and its components, which are called organelles, and it divides its cytoplasm, splits in exactly half, similar to what we describe in cell division, our own human cellular division, that is mitosis, not meiosis, but mitosis. Now, it's asexual reproduction. In other words, they don't need other, the cells just simply make copies of themselves. They don't need another cell involved in order to replicate itself. There's no involvement of other cells. They just simply replicate themselves, reproduce themselves by dividing their cytoplasm, dividing or duplicating their organelles, and duplicating their DNA. Now, the bacteria are named based on genus, or genus, however you want to pronounce it, and species. So genus is the family name, and we, in the scientific world, came up with a way to categorize and a way to have the nomenclature set. So the name will be capitalized. italicized. The groups of the bacteria with similar characteristics are italicized, but the name is capitalized, all right? The species is the second name. So you know how we have the first name of the individual and then the family name. Well, you can consider the second name the family name, which is basically your family is the species that you come from, right? So think of Homo sapiens, right? That's us. So the species is sapiens. So a species is the second name. It's going to be lowercase. It's going to be italicized, and it represents the specific characteristics of a particular group of bacteria. So that's how we categorize, identify, and name bacteria. So bacteria can be anucleates. In other words, they don't have, they are anucleate. I shouldn't say can be. They are anucleate, but they do carry what's similar to a nucleus. It's called a nucleoid, which stores the DNA. And then, of course, there's a cell wall that's called the glycocalyx, and some of them have a slimy cell wall to prevent, the killing of it. So it does play a part in resistance to certain antibacterial agents like antibiotics. All right. What I want to mention also is that most bacteria will not have that much locomotion. Others are very movement oriented with cilia and flagella. Most do not have spores, but some do have spores, and the spores are dangerous for us because we can inhale them. They can get into our body and lay latent for years. These are inactive bacteria that are very difficult to kill. And when they activate, they come out of their spore and do their damage. So tuberculosis is like that. And I mentioned that in lesson 1. Now, let's go over the 3 basic shapes. So make sure you understand the 3 basic shapes of bacteria. It's very simple. There are only 3. It's not complex, so please remember this. So you have cocci. Don't pronounce it cockeye. It's cocci. They're spherical round bacteria. They can be together in clusters, or they could be in chains. We also have rod-shaped bacteria that we call bacilli, and we have spiral bacteria that kind of look like the pasta that we buy that are spiral shaped. Those are called spirella. Okay, so these are the 3 main basic shapes, and you're going to see the most common bacteria that we name Staphylococcus aureus. Well, Staphylococcus, you already know that these are spherical bacteria. They are found on your skin. And if they are out of balance and there is overgrowth, you can get a Staphylococcus infection. Very common nosocomial infection in the hospital. All right, so cocci can be mono, diplo, strepto, or staphylo. So mono means one single cocci. Diplo means paired. Strepto means a chain. And Staphylococcus aureus, which is the most common bacteria on your skin, that means it's clustered. All right, so this is terminology that you need to learn and have it down pat, because you're going to see it over and over. It is the foundation of microbiology, and you'll be using it a lot in medical microbiology. Now, bacilli, as we mentioned earlier, are rod-shaped. They may possess flagella. They can be present in pairs. So those are called diplobacillus. Bacilli is plural from Latin. Bacillus is singular. We can have chains, which are called streptobacillus. All right, so one chain is streptobacillus, many chains, streptobacilli. We can have E. coli, which is a rod-shaped bacteria. This is normal flora of the intestinal tract. But if it happens to migrate, maybe perhaps poor hygiene or poor wiping, if it migrates to the urinary tract, it will cause a urinary tract infection. If it leads to the bloodstream, it can cause bacteremia, sepsis, and death. All right, so not to scare you, but that's the sequela of this infection if it gets out of control. Of course, we have antibiotics for it, so most people don't get to that level. Now, bacilli will and may form spores. Not all of them, some will form spores, so just be aware of that. All right, the next one is spirella. These are the spirochetes. They are spiral shaped. You'll note, we're going to talk about gram-negative and gram-positive very soon. It has to do with a staining technique. And if it stains a certain color, it's gram-positive. If it stains another color, it's gram-negative. Gram-negative will stain pink. We're going to learn that, don't worry. But just remember that the spirochetes are gram-negative bacteria. They're spiral-shaped. And examples of that is the disease syphilis and Lyme disease, which is caught by Borrelia burgdorferi, which is a bacteria, and it is transmitted through a tick biting a human, and the tick comes from a vector, the deer. So it is boom, 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 boom. So it just kind of hops along down to the human being, from deer to tick, from tick to the bacteria Borrelia burgdorferi, and then of course the spirochete enters the body and causes havoc, joint disease, and that's Lyme disease. All right, so all of the bacteria that we can identify that causes infection, as you know, they're pathogenic. These bacteria that are truly virulent, they must first be identified in a lab. So in order to do that, you must collect the specimen properly, keep them alive enough for the culture to grow so that we can be able to identify the culture and then treat it with an antibiotic and do culture insensitivity reports that can go back to the practitioner, the clinician, so that we can determine which is the best antibiotic for the patient. Meanwhile, we don't want the patient to die. We give them a broad spectrum antibiotic, empirical therapy based on deduction and theoretical understanding that based on the clinical view of the infection, as well as knowledge of simple things like certain bacteria smell a particular smell, like Pseudomonas aeruginosa is caught as a bacterial infection that's often due to being in a pool too long, contamination of that bacteria, has a fruity odor. Bacteria that have a foul smell or anaerobic. So you can determine empiric therapy until you get the final culture and sensitivity report. So in order to get a culture and sensitivity report, you must be that collector that collects it properly. So there's that. So this is the route of the technician. You're drawing the blood, you're obtaining a culture from a wound. Perhaps the nurse is doing it, perhaps an infectious disease specialist is doing, whatever the case may be, you need to collect it before administering the antibiotics. Collecting without contaminating the specimen is key. So there's technique in doing that, and not touching other surfaces, not touching the skin, but going directly to the wound. There is a method to the madness. There's a protocol. There's a talent involved. There's definite a learning curve to doing acquisition of collecting the specimen. So place the specimen in appropriate container. Again, you can contaminate the containers, so being careful with that, and then transporting the media properly without extreme temperature, because you don't want the bacteria to die. You want it to grow so that you can apply the antibiotic discs, and then those antibiotic discs will show the zone of inhibition, and therefore you can determine the larger zone of inhibition is good, because that means that that antibiotic is really killing the culture. All right, so guidelines for specimen collection is obviously labeling. I mean, that's the obvious, labeling the specimen properly, transporting to a lab in the allotted time so that the bacteria don't die, no extreme temperature, no extended time. The safe collection provides accurate results. Handling the culture when you're going in with the sterile cotton tube into the wound, right out and into the media, all right, no contamination, provides accurate results, all right. Prevention of the spread of the particular disease to other healthcare workers or patients. Make sure you wash your hands, make sure you're gloved. All of these things are super important when you're doing a wound culture per se. All right. There's also blood cultures that we do. So, there's a lot to think about when it comes to medical microbiology. Now, bacterial staining. The staining involves different dyes that we utilize that will improve the visual identification of the bacteria on a slide that is prepped under the microscope. The color will lead to identification of bacteria, and it provides the classification of the bacteria. If it stains pink, it's gram negative. If it stains purple, it's gram positive. So simple stains will illustrate the structure and the arrangement of the bacterial cells, and the differential stains will also supply information of the bacterial walls. So the gram stain technique we'll go over later on, but suffice to say, you need to read the chapter in detail. It involves using this stain that's called crystal violet, which will stain the bacteria purple. You wash it out with water. You wash the slide with water, and then you stain again with iodine, grams iodine, to assure the adherence of the bacteria on that slide. And then again, you apply alcohol to decolorize the rinse, so that the only thing that remains stained is the bacteria and not the entire slide. And that's how we come up with isolating the bacteria purple, those that are gram positive and will connect with that purple, chemically bind with that purple stain of the crystal violet. And that's the easiest way I can explain the gram positive stain. So the bacteria that retain the purple color in the end are gram positive. And then what you do is you want to make sure that they're not gram negative. So to do that, next step is, you're not done. You're going to take that slide, all right, and you're going to counterstain with a red dye called safranin. And so now the bacterial cultures there that stain with red or pink are going to be gram negative. So this way, you look at an entire wound culture, you're going to see gram negative and gram positive bacteria, and then you're going to see which are the ones that are dominating, and then you can identify based on using antibiotic discs how to treat it, all right? Which is the dominant culture that is causing the infection? Is it gram positive? Is it gram negative? And then using the antibiotic discs to treat it and finding the appropriate antibiotic for it. That's called culture and sensitivity report. And so the report comes back to the physician, back to the nurse, and then obviously determining the course of action, because probably until that culture and sensitivity report came back, the patient was receiving empirical antibiotics, but now we can really hone in on the bullet that kills the bacteria, and meaning the bullet being the antibiotic. So bacteria retaining the red or pink are going to be gram-negative, and bacteria that retain the purple are going to be gram-positive. Just take this as a mental note that the ones that are gram-negative, honestly, you don't want to have a lot of gram-negative. Those are the gram-negatives that are going to have a tough cell wall. So if they stain pink, know that they have a tough cell wall, and you got your hands full with the gram-negative infection. All right, now we also have another kind of stain. It's called the Zeal-Nielsen stain. It's called the Zeal-Nielsen stain, but the more popular pronunciation is the acid-fast. This is the common term that we use because, although Zeal-Nielsen discovered how to utilize this stain, we prefer to just use the acid-fast terminology because it describes how we go through the staining process. So the acid-fast stain is used specifically on Mycobacterium, meaning these are the causative agents of tuberculosis and the causative agent of leprosy. So the acid-fast organisms are going to resist staining due to a glycolipid and mycolic acid layer. That's why they're called Mycobacterium. So these are very strong bacteria, TB and leprosy. And the only way to identify them is through the acid-fast bacteria, which will be able to retain the stain. So acid-fast bacteria retain that carbofusion stain, and they will appear red against a blue background. And that is a sign that the person either has TB or leprosy, some kind of mycobacterium. All right, so then there's also culture insensitivity, which I've been talking about, which is once you have identified the specific specimen, you inoculate into the proper culture media that have the nutrients to allow for growth. You want the microbes to grow. You place it in an incubator, you allow them to grow under proper temperature and humidity, you let them grow, let them grow, let them grow. And once the cultures grow, then you test these cultures with different antibiotic discs to determine the best treatment option for killing that culture. So that way you know how to kill the bacteria inside the body of that patient. All right. So the cultures will thrive in liquid broth, semi-solid, or solid agar. So what are these? These are nutrients and just a petri dish that contains wonderful nutrients that allow the bacteria to grow. So you want to use a sterile petri dish that has been obviously unused. You open it from a package. It's clear. And so then you do this Z kind of swab with the Q-tip that you use from the culture, from a wound, for example, all right? And you zigzag across the entire disc. And so by doing that, you spread the bacteria, streaking it across the disc in a pattern that is going to cover all four quadrants. So you're going to go zigzag, 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 turn it, zigzag, 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 turn it, zigzag, zigzag, zigzag. And then the fourth time, another zigzag, you get the point. It's all four quadrants are involved. That's how you do the culture, spreading it across this petri dish. Then you put it bottoms up in the incubator so that there's no dripping or condensation onto the colonies. That's super important. All right, bottoms up. You don't want the condensation killing the colonies, so bottoms up. All right. So the agar and the colonies are facing up, and so this way there's no condensation. All right. Now, let's talk about the culturing. You could do the primary culture, usually available probably between 24 to 48 hours after the incubation period. You can have a mixed culture, which is identifying more than one microorganism. And at that point, you have to separate to yield the pure culture of only one species. And then after isolation of all the microbes, you examine each one to identify the microbes, right? Meaning you examine each culture to identify the specific pathogens. All right. So the pathogens identified under microscope will be identified based on appearance. Also, the growth requirements on that agar, the biochemical tests that can be done, a DNA analysis, and all those four components may require 7 days to finally figure out exactly what is the culprit, what is the pathogen. So after identification of the microbes, you can determine the treatment via the disc diffusion, as I've been mentioning. This is called the Bauer-Kirby test, where you have the antibiotic discs, which will provide the zone of inhibition. All right. At this point, you need to do a broth dilution, which is a quantitative analysis, which identifies the concentration of drug amount needed to kill. So this is called MIC, the minimum inhibition concentration. This is, again, MIC, minimum inhibition concentration. It identifies the absolute concentration of drug needed, the minimum drug dose needed to kill the culture. So the minimum inhibitory concentration does not identify if the organism is killed. So you can have the minimum bactericidal concentration, which will determine whether the organism is killed or just inhibited. And of course, you do the testing with FDA-approved machines that may be expensive and unable to detect certain resistant bacteria. So it's easy to just go on with that Kirby-Bauer test that I mentioned. The treatment begins with culture and sensitivity report from the Kirby-Bauer test, doing the antibiotic disc and finding the zone of inhibition. All right, so that's the best way to be cheap and effective in treating the infection or suspected infection. The usual bacteria can be noted with a type of infection based on smell, based on look of the wound, and of course, based on the initial culture and sensitivity report. All right, so bacteria that cause disease will destroy tissue and causes ulcerations and cellulitis. Some really damaging bacteria can cause the release of endotoxins from their cells. These are found in gram-negative bacteria, and also 1 gram positive called Listeria. And then there are bacteria that have exotoxins. For example, gram-positive and gram-negative bacteria, some of them do have exotoxin release. So bacterial infections will cause high fever, will cause a high white blood cell count with a shift of neutrophils elevated. If the infection is severe, it may cause pain at the wound site, tachycardia, which is rapid heart rate, tachypnea, rapid breathing, foul-smelling drainage from the wound, and IV antibiotic therapy is the only way to go, possibly removal of the abscess through incision and drainage, debridement, and possible amputation. The only way to kill initially is antibiotic therapy, but surgical intervention if the antibiotic therapy is failing, whether it's empirical or even sometimes you've done the culture and sensitivity and you have an IV antibiotic that you know is killing the culture, but the bacteria is spreading faster than the antibiotic can work. And that's when you need to do both IV antibiotics as well as incision and drainage of an abscess. And if there's necrotic tissue, debridement. And then, of course, if there's any kind of cell death and tissue death, where there's complete gangrenous changes, amputation. I'm very familiar with this because this was my forte as a podiatric surgeon. This is what I did: diabetic foot infections, managing wounds and amputating. All right, so antibiotic classification, you can have the antibiotics that are going to kill the cell wall. Those are bactericidal agents. Those are the ones that are really good. Those are the ones you want to take. But sometimes you can't take those for whatever reason. Sometimes it's allergies. Sometimes that particular bacteria that has infected the individual, you only have bacteriostatic agents for. All right, so bacteriostatic is going to diminish the replication of the DNA, which means that eventually the bacteria will be killed because they can't replicate. Bactericidal will kill the cell wall and kill the actual bacteria right away. Broad spectrum means it kills both gram-positive, gram-negative, and others. Narrow spectrum is what you want if you already know what the infection is all about. You don't want narrow spectrum if you don't know what the antibiotic or what the wound is carrying. If you don't know what the wound is carrying, you don't do a narrow spectrum until you get the culture and sensitivity report back. You do a broad spectrum, get the report, culture and sensitivity report back through the Bauer-Kirby test, and then you can figure out narrow spectrum antibiotic to kill specifically the bacteria in that wound. All right. Mixed wound is going to have both aerobic and anaerobic bacteria. And God help you if you have superbacteria, which are resistant. For example, methicillin resistant Staph aureus, MRSA, very common. It started out as nosocomial infections. It's very common now to get this infection. And we have antibiotics against MRSA. And then, of course, those antibiotics became resistant. We used to have vancomycin, and now we even have vancomycin resistance. And we use Cubicin, which is like $3,000 IV a day. But we keep finding different antibiotics as resistant strains come in. We call those superbacteria. We're due for some superbacteria strains coming up because we have overused antibiotics during the COVID-19 pandemic to deal with the virus. And then, of course, patients were so immunocompromised from the virus that they had superseding bacterial infections. And then doctors would, prescribe antibiotic therapy for, bronchitis, which is the taking over of bacteria from an initial viral illness. Okay. And so that's how we have come to superbacteria. So to summarize this lesson today, we did a lot. We discussed the field of microbiology and how it's relevant to medical microbiology. We discussed the two names on how we identify organisms by genus and species. Capitalizing genus, species is italicized. Then identifying bacteria based on three shapes: cocci, bacilli, and spirella. And then to circle back and summarize, we talked about how it is extremely important in caring, how we access the wound or blood culture and transporting the actual specimen to the lab and then growing the specimen in the lab, and then the staining procedures, the identification of the bacteria, determining bacterial susceptibility to the Kirby-Bauer test via the minimum inhibition concentration, utilizing the antibiotic discs, and then, of course, determining antibiotic therapy. All right, so that was my lesson for today. I do hope you enjoyed this lesson, and I will see you next time. Again, if you have any questions, feel free to contact me, email me, and good luck studying. All right, bye-bye.